Okay. So first of all, thanks to the organizers for setting up this wonderful meeting. And now it becomes a little more applied. So I would like to show you how we connect the neuronal imaging with behavioral data. So we use a not very common animal model, namely the honeybee, and I'm going to start by motivating this. Then I show you some of our methods, and finally I'd like to present two applications, namely what do we learn about the representation of odors in the brain of the bee, and whether we can detect signs of sleep in this animal model. Okay, so why bees? Because they have a mini brain of just 1 million neurons, and this brain is also of very small dimensions, so 1 millimeter cubed. And if you know the penetration depth of two-photon microscopy, that simply means that we have optical access to almost the entire brain. But besides these practical advantages, these bees offer also a very rich behavioral spectrum. So they are excellent learners. They navigate extremely well. They are communicating. They outperform many of the mammals with their communication languages. They live in social organizations. So by itself, it's already a very interesting model to study, but also has what we think a good potential of translational research. But if you look at that brain, that doesn't really look like a mammalian brain. But some sub-networks of this brain actually are very similar. For example, the olfactory network. What's the olfactory bulb in the mammalian brain is the antenna lobe in the insect brain, and this is basically a one-to-one copy, just or apart from the dimensions. So receptors are sitting here on the antennas. They, once activated, forward this activation into this antenna lobe network. Each of these nodes here, called glomeruli, receive input from a single receptor family, and they are interconnected, each of these nodes to each of the others, and this processing step is then forwarded into higher brain centers. And today I'm going to talk only about that first processing step. So now, with two-photon microscopy, we heard we can easily do three-dimensional imaging. If you want to now know something about the structure of this antenna lobe, we stain it with anti-synapsine and then optically slice it via two-photon microscopy, and then we are able to reconstruct a three-dimensional image. And this serves as our atlas, which we then use if we do experiment in different subjects to identify these single nodes. Now, we would like also to image functions, so we have to stimulate this olfactory network with odors, and that we do by home-built olfactometer. So you see our objective here, a plexiglass block. You hardly see the bee sitting here below, and that olfactometer with this nozzle here is pointing directly at the antennas. That works here with 8 different odor channels. Each of this switches between an odor-filled vial and an empty vial, and this we need because at the end the output flux here is always the same, independent of which of the channels we switch to odor. So we have now the possibility to have 8 single odor components or an arbitrary mixture of those. And important for us is also the temporal resolution, as you will see later. So it's important that these valves here switch very fast, and that the stimulus arriving at the animal is very well defined in time. So a 5 millisecond resolution is what we are getting from this device. So now, if we want to do functional imaging, we use a calcium sensor that detects neural activation in the neurons, but in honeybees we can not just genetically express this, because transgenic bees do not exist yet. So we have to manually insert this sensor by very small capillary with micrometer size, hitting the axons of the neurons that we are interested in. And if we are successful with that, the dye diffuses into the neurons, and what we are going to see then, if we image again with two-photon microscopy, is the marker is changing fluorescence when these single glomeruli, so the nodes of the antennal lobe, are activated here or are inhibited there. And if we repeat this experiment several times, we see that this pattern always looks the same. So for a single odor, this is well conserved and represents this odor in the antennal lobe for a single animal. Now we would like to do experiments with more subjects to understand whether this is a universal code. So we have to identify these glomeruli. And we do that, for example, by a regional homogeneity analysis. So we look for the correlation of every of the functional signal here with its neighbors. And we find that correlation being very high within these nodes and decaying strongly at the edges of these nodes. So in this way, they are well, there's a good contrast between these single nodes. We look at our atlas and we give each of these glomeruli a number. So then these videos we can acquire with a 10 Hertz frame rate. But for some of these experiments, this is not enough. So we have the possibility to go 1D and define a customized scan line that is passing through all the glomeruli of interest, and that we can run with 200 Hertz. And then we get these very well resolved response maps in time. So for different glomeruli, different odors, we see a map of different response patterns. And now we would like to interpret this and to understand this. So first of all, what we see is that if we average over the activity that is elicited by the odors, we are getting a pattern that is well resolved across subjects. So here we do an experiment in 25 bees. We are stimulating always with the same odor, 1-hexanol in this case, and we find for the 20 glomeruli that we are testing here a very similar pattern. All the time, 3 of these glomeruli are activated. And this is nothing new. This has been found 20 years ago already with wide field calcium imaging. But what can we do now that goes beyond that? Two-photon microscopy now allows us to not just look at average activity, but to time resolve this activity. And what we see here is now the average over the subject, but the time dependent pattern. So at 0 here, we are starting our odor pulse, and it stops at 1 second, and we now wanted to see whether there's some information in that time behavior. And the first thing that caught our attention is that it seems that there is a different offset when glomeruli start firing. And this is something we wanted to understand. We wanted to understand, is this also odor specific? Does this maybe contain already information on the odor? And for that, what we do now, we kind of need to normalize this data because different subjects always show a little bit of a jitter. So what we do is we forget about all the absolute times here, and we simply rank the glomeruli in the order at which they start firing. So we do that for these averaged results, and we get what we call latency rank templates. For each of the order, we have just an ordered number of glomeruli in the order they are activated. And we would like to know now, is this simply an arbitrary order that we find here, or is this something that repeats like that in each of the single experiments? So we now compare for the single subjects the ordered vectors with that general template that we get from the average data. And we would like to know, comparing correlations, does one odor always correlate better with the correct odor in the general set, or is that simply arbitrary across all these templates? And this is what we find. Indeed, the latency code is well representing already the odor, so we get highest correlation always for the correct template. That is interesting. So it doesn't need an average value. The brain doesn't have to wait until an average firing rate is established. It seems to know at the onset of the odor already enough information to identify this. However, you see small deviations here. For example, between 1 hexanol and 3 hexanol, this latency code is not so robust. It seems to mix those two odors, which is not a surprise because they are chemically extremely similar. So this is a 10 carbon chain, and here the hydroxyl group is sitting at the first carbon, here at the third carbon. So likely the smell of these components is also very similar to bees. Now what we would like to do is to understand how does this latency code compare to the amplitude code, which has believed until now to contain most of the odor information. So we do the same thing. We create vectors from the average amplitudes. We look at the correlation of single experiments to the average amplitude vector, and this is what we get. So extremely similar confusion matrix, so it doesn't perform much better than the latency. For some odors it does. For other odors, even the latency seems to be more specific for the odor. One question now remains: is this actually independent information, or is this completely redundant? Because one could also it could be possible that if the amplitude is high, that the activity will also raise earlier. So this could be representations of exactly the same amount of information, and then we wouldn't gain anything by looking at latencies. So to test this, we now create vectors that actually contain both of the information, latency part and the amplitude part, and then we do the same correlation to understand, is this now representing odors more specifically? And indeed it does. So we have in the combined code an even stronger odor specificity that mixing but still we also, as for the amplitude, get here the confusion in the 1 and 3 hexanol. But generally we can say from this that there must be information in the latency that is not in the amplitude and vice versa, because that combined code performs better, and we can quantify this by averaging now across the diagonal. So we get a general prediction accuracy that in the latency code seems to be comparable than in the amplitude code. But if we combine these data, we get significantly better prediction accuracy than if we just took the single components. So far, this is something that we, how we interpret the response pattern in the periphery of the brain. Now, the question is, does the bee actually use this information as we think it could? So we have to connect these data somehow to behavior. And for that, we have a very interesting paradigm, the proboscis extension reflex. If that bee gets in contact with a sugary solution, so for example, a floral nectar, it extends its proboscis. And we can use that now for a conditioning experiment. So classical Pavlovian conditioning, we superimpose an odor that comes from this nozzle here with a reward, a drop of sugar, and we give that slightly afterward with a small overlap. So because bees are excellent learners, after 2-3 trials, the bees knows how to connect the odor to the reward and extends its proboscis already when only the odor comes in awaiting of the reward. This you see here. Oops. When the odor is arriving, you see a signal here, and you will see the bee extending its proboscis here, and because it awaits the reward. So such a training session lasts 5 trials, then the bee has learned that and memorizes this for the rest of its life. So now what we do after we are teaching now 5 groups to the 5 different odors that we had used in this experiment, and then we test each of these groups, not just against the odor it has learned, but against all the odors to understand, does it distinguish between those? And that behavioral generalization matrix looks like this. So extremely similar to what we have found in the periphery with that confusion feature here. That does not mean that the bee is not able to distinguish between that odors. It simply means that the difference is so subtle that it doesn't care. It is hoping to receive the reward in any of these cases. So we see very nice correspondence of both codes. Here there is a small difference, but also this we think we can interpret because this is isoamyl acetate, which is a component of the bee alarm pheromone. So usually a stimulus with a negative value, and now we are connecting this with a positive reward. So in the higher order brain centers, probably the bee gets extremely confused, and therefore we see this confusion here, which is not present in the periphery. But otherwise, we see how that response pattern in the periphery seems to be transported very well conserved to the behavioral level. Okay, now let me rush to the second experiment I would like to show you, and that is we want simply to test, do we see bees sleeping, and are we able to see a representation, a neuronal correlate for sleep in the brain. So you see again a bee under our two-photon microscope. Here's the objective, the head of the bee, the antennas, the abdomen. Now we point a camera on that bee, and yeah, what's a signature of sleep? Mechanical motion. So we want to understand, does it change its motion? And for that we use simply the variance of the camera pixels. So for each of the pixels, we simply measure how does that change in time, and then if the bee moves strongly, we get such a variance image. If it is resting, we get this one. Now we run this experiment for a long time, and we have to do that during the night because bees have a circadian rhythm, so we have to fully automate this experiment. So then in real time we analyze this variance, and then astonishingly we see this quasi-binary switching. So there is always periods with quasi an identical movement strength, and then it goes down, and again the variance seems to be very similar to a single value here. So this already suggests that there may be two different states here. So we have to automatize our neural imaging, so we trigger our microscope whenever we detect a switching of the variance here, and we take images of 1 minute, and if we are lucky, then we have a night where we find so nicely samples from both of these states that we can then analyze. So now, analysis works the same way. Raw fluorescence, regional homogeneity analysis to identify the single nodes here. Then we average intensity over these nodes, and what we get here are these response time series for each of the glomeruli here, in this case 12. So I show you one example here of each of the states. Here an active bee, here a resting, or the same bee in an active state and in a resting state. And so we are not stimulating, this is spontaneous activity. So when we saw that for the first time, we thought, oh my God, there's absolutely no difference. So what do we, shall we continue this experiment? But we searched help then from a machine learning algorithm, and astonishingly, it has absolutely no problem to classify between rest and awake states. So here you see a classification success of 93%. So attention, because this data that I'm presenting here now is very preliminary. So this is a single subject. We have so far very little data, no statistical significance yet, so this has to be taken with caution. But now what we wanted to understand is, in this huge success rate, which features of our response maps are actually contributing to these differences in the state, because our exploratory data analysis didn't show that at all. So to understand this, what we used is a random forest classifier, which is using not always all the features that you feed it to search for a difference, but a random subset of features. And then later you can look in the cases where the right state was classified, which of the features have actually contributed in that cases to that correct classification. And interestingly, we find that it's not properties of statistical properties of the single time series, but it's rather the connectivity, the network properties that are allowing for this distinction. So we looked at the networks, and this is what we find. So here, 3 different subjects, awake state, sleep state, and what we see here is only the significant connections between the glomeruli. So we scramble the data, form surrogates, and then we test significance, and this is what is left in connections. And now, interestingly, what seems to happen when the bee is falling asleep, all the connections in that network seem to break up. Some new connections are formed, but the general connectivity, so here this is Pearson's R, is dropping drastically. And that is very interesting to us because this is somehow what we also know from human sleep, that the receptors still receive input, but the data is not integrated anymore in the brain. And this is what happens here as well. Yeah? So we have activity in all the single nodes, but it is not connected. This network is not connected and is not forwarding the usual code. So this gives hope that we indeed also with this simple animal model we will be able to study mechanisms such as sleep. So yeah, my time is over. I would like to thank all the people in my laboratories. The ones that I marked in yellow here are were directly involved in the applications that I showed you here. Thank you.